from the National Human Genome Research Institute in Bethesda, Maryland. We're been working on GNE Myopathy for some years towards in our natural history study and a few ManNAc trial. We're currently conducting a third ManNAc trial right now together with other centers. So let's start with the natural history. And on this, Michaela made a good plea for the need for natural history earlier yesterday. So we've been having the natural history since 2011 at the NIH Clinical Center. That's where we are. To conduct the study, basically, we have 4-day admissions, and I'll have a sample here of what the kind of schedule is. Where we try to study as much as possible, both function, both the muscle itself with the muscle MRI, but we do a lot of evaluation and rehabilitation medicine with physiatry, physiotherapy, occupational therapy, as well as a thorough neurologic evaluation. We collect research samples that allows us to have a repository of samples of plasmas and cells that can be then used for experiments and collaborations when we have them. Overall, we had 69 patients that were recruited. 56 came to the NIH Clinical Center, some for only one visit, some for several visits, and 13 were enrolled on a part that was for questionnaire development only, which I won't go into details. Dr. Lockmuller presented some similar data from the registry yesterday, but basically we see a little bit the same, where there's a 7 to 9 years of diagnostic delay. So there's certainly some improvement there. We've collected which kind of diagnoses were given to different individuals in our natural history. We can see that up to a fourth had the initial diagnosis of limb girdle muscular dystrophy, and then a lot had some kind of neurological neuropathy first diagnosed instead of a myopathy, which led to several attempts of treatments that were not necessarily indicated in their condition. So we had quite a spread there. About 72% of a cohort received another diagnosis before receiving the one of GNE myopathy. We've also described several new variants throughout in the natural history. We've tried to collect as well data from the literature to analyze those variants as much as possible, understand them. We conducted several other experiments in the lab to try to further prove some of the variants. It's been a difficult task because there's many unique variants. There's a lot of missense variants, so those changes that don't interrupt the protein fully, but just change one amino acid for another. So one building block for another. And we know that those changes with our current guidelines typically require a little bit more work to be able to be defined as definitely pathogenic. Most often we know that it's very likely the right answer, but for trials and so on and so forth, we need to be able to prove to regulators and everyone that we really know what we're doing. We really know those variants, and there's a lot of work that we've put into trying to understand better the variant burden in GNE. One of the main tools that we've used in the natural history is called the quantitative muscle assessment system. So it is a system of fixed frame dynamometry for maximum isovolumetric muscle contraction. So basically it means that there's a fixed frame, and we're testing the muscle strength without moving. So the limb will be attached with a strap, and there's a little box that registers how much strength is applied. And then when applying, the frame doesn't move, and we get that recording in there. So that's been the method that we've been using. In the natural history, we worked on 13 different muscle groups that we've collected longitudinally. In further studies, we've used the 6 core muscle groups that we get better results. And when we look at this data, it looks really like a cloud of different dots. So when we try to correlate it with age, we see that there's a wide variation. And anyone that has worked with GNE will probably say the same of oh, we know that some people have symptoms much earlier and some much later, and we see that kind of variation with age. So for one particular person, so if we hear A, B, C, D's are different people, we see that the pattern is going down from the first measurement we have towards the last ones, but from the same age, we have a wide variation from completely normal to absent contraction that we can measure on QMA. So we were trying to find other ways than correlate it with age. And that's where the GNE disease progression model came from. So the goal of it was to try to normalize everyone, to be able to compare everyone to each other, to have outcome measures for future trials. So what the model does is that once the knee flexion movement reaches 50% of what we predict, we define this as the disease age 0, and then normalize everything compared to that. So we take into account the age, the timing, the rate of muscle involvement, the muscle specific variability, and we get those curves where we can see that, for example, if I center it at 0 disease age and 50%, that's the horizontal line, we can see that the red line is the ankle dorsiflexion about 7 to 8 years before we reach 50% of the predicted strength. And then shoulder abduction, which is in blue, comes about 2.5 years after we reach 50% for the knee flexion, we reach 50% for the shoulder abduction. So we get those patterns that allows us to predict a little bit better what we expect should happen in the disease progression. And when we compare it to all the rest of the data that we accumulated in the natural history, so if we go on composite strengths, like in both the upper limbs and lower limbs, the 6-minute walk test, the AMAT, which is the adult myopathy assessment test, which people have to do modified push-ups, have sit-ups, there's neck strength, leg strength, different tasks there. If we look at the human activity profile, which is a questionnaire that asks which activities can be done, it cannot be done anymore, or the inclusion body miles, that is functional rating scales, that's a 10-item scale with different functional elements. All of them correlate better with disease age than with chronological age or even disease duration. If we take the first onset of symptom to the point that it was, we do see some correlations with both, but disease age correlates much more highly and consistently with all those parameters. So it's felt that it's a better outcome measure for us to try to look at. And that's some examples. So we have here someone that's at disease age 0, and we can see here it's on the IBM-FRS, it's different items. So we have dressing, we have handling utensils, climbing stairs, walking, so on and so forth. And as disease progress and disease age progress, we see that the answers the scores go down on the IBM-FRS. The other thing that was included in the model is a treatment parameter that we call the gamma parameter inside of it, which allows us to see how a treatment affects the trajectory of the disease. So we don't necessarily expect something linear. We can see that there's some curve in there, but if we're really early in the onset of the disease, a gamma of 0 means we would have completely stopped the progression at that point, versus a gamma of 1, we are completely on the curve and the treatment has not done any effect. But so the earlier, the better. If we get very late, we may not be able to see effects. The point I want to make with this is that for trials, we have the difficult task of saying, yes, we know that earlier the better, but we kind of have that challenge of when we're really early in the disease, we can kind of see that we don't have, inside of a couple of years, a lot of distinction between the lines. They're still very tightly packed. So the middle model, where we are really in the highest slope, is where we targeted for our current study, to be able to make the most difference, to see more difference within a limited period of time. Because we cannot have trials for 10 years, 15 years. We know that there's a limited amount of time, so we targeted this. We've also looked at other outcome measures. So we looked at SNA lectin staining that was helpful in our phase 2 trial, where we measure sialation on the surface of muscle cells. We've looked at muscle MRI, and we see that the degree of fatty replacement correlates with various other outcome measures, and we've done spectrometry on this. So other outcome measures are there. They've been used. Some of them have been used in previous phases. Some of them have been developed since. And then I'll go in the meat of the ManNAc trials. So we've conducted 2 ManNAc trials at NIH. ManNAc has been identified, and we've heard this from previous speakers, as a potential therapy going really in the middle of the pathway. So we're really here in between the 2 functions of GNE. We are not sure of the method by which ManNAc gets phosphorylated. There's all kinds of theories, but we know that it gets to sialic acid eventually, and we've demonstrated that in both the phase 1 and phase 2. And that's some of the data of phase 1. So the phase 1 was really a single dose of ManNAc or placebo given. So we had 3 different doses of ManNAc, either 3, 6, or 10 gram, which are here noted as in thousands of milligrams. So 2,000 milligrams, so on and so forth. And we can see that with a single dose of ManNAc, first of all, overall, it was well tolerated. The 10 gram was had a lot of probably unabsorbed ManNAc. We've talked about the absorption of ManNAc sometimes being an issue. So we had more diarrhea and nausea. In the 3 and 6 grams, we didn't see as many on the single dose study. We saw that the ManNAc concentration would peak at about 2.5 hours, but the sialic acid response peaked at about 8 to 11 hours and was still present at the end of the 48 hours we were collecting samples. So we see after administration of ManNAc a sustained response of the cells in producing sialic acid that we can detect, which is very important for us because sialic acid by itself is known to have a very short half-life. So we know that this sustained response 48 hours later is not only the circulating sialic acid, it should have been cleared by then. So it's really a response to the intracellular intake. Given the model, we can see that the ManNAc peaks better with 10 gram, but the response of sialic acid doesn't peak as well. It stays pretty much the same with 10 gram than 6 gram. That suggested that 6 gram twice daily was a better dosing there. So we went forward with a phase 2 that was conducted, if I remember correctly, the 2015 to 2017. We've seen 12 individuals that were randomized initially to either 3 grams or 6 grams twice a day of ManNAc. And then after 7 days, everyone was put on 6 grams. The primary outcome measure in there was muscle biopsy. So we were collecting muscle biopsies, one next to each other in 2 different limbs to see S-lectin staining. So fairly, not a minimal intervention to be able to demonstrate, but we were able to get good results with that to show that really the muscles, which are what we're interested in affecting, did indeed have a response. And then we looked in the study extension more into clinical outcome measures. Bear in mind that these, with 12, we were not expecting necessarily to be able to demonstrate a difference on the clinical outcomes because it's a very low number. But overall, we can see that there's an increase in muscle sialation after administration of ManNAc. So on the left hand side, we have a muscle biopsy at baseline, and then after 90 days, we have the one on the right hand side. And the green is the S-lectin staining, and the red is just a marker, caveolin 3, just to see the contour of the muscle cells. So it's kind of our controls. And when we compare both groups, we can see that between baseline and day 90, we get an increase in the S-lectin signal that we can quantify it even when we normalize to our control, which is the caveolin 3, we still see a robust increase overall in the cohort. So it seems like the ManNAc administration does what we want it to do, is that we want more sialic acid on the surface of the muscular cells, and that's what we're getting. Another thing that we were interested in is we're giving ManNAc. We know that we're in between the 2 functions of GNE. So is there some kind of genotype, phenotype? Like we're not necessarily in mice, so we cannot see necessarily expect the same response that we've seen in preclinical trials. So we've looked whether people had kinase variants or epimerase domain variants and tried to see if there was a difference in the response, and the answer is no. So basically, whatever the variants that were observed, whether they were in the epimerase domain or the kinase domain, we see still a response by ManNAc administration with an increase of sialic acid, which is what we had seen in the preclinical models, but we wanted to make sure that we had the same kind of response once we went in humans. On the study extension, when we looked at different parameters, we had some encouraging signals. So when we looked at lower extremity strength, upper extremity strength, we had pretty much not much change from baseline. And then when we compare to the natural history, we can really see a difference in between the two cohorts. Sorry, we've lost some data at months 24 and 30 in the upper limbs because of an issue with one of the devices that was used, and that really made some issues when we were looking at the gamma parameter. But we can see as well on the AMAT that there was some difference as well. So despite the fact that we didn't have the most robust cohort for the clinical outcomes, we were still seeing some changes that were encouraging. And same when we looked at the gamma parameter that I talked earlier. So at 12 and 18 months, we see that here we see the range of the gamma. If it overlaps one, it means there was no change. We see that we're towards the 40% slowing in disease progression at 12 months and 18 months. Once we lost the hand grip at 24 and 30 months, we encountered more problems. So we've been very careful with the monitoring of the QMA system for this new study, which is the NN109 Medgene trial. We'll talk shortly about it. It's still ongoing, so we don't have data to share on, and it's blinded, so we really don't have any results that I could say, here's what we're observing. But it is a collaboration between 3 of the NIH institutes, a pharmaceutical company, Ladiant, and Neuronex, which has a network of other sites. So in between the NIH Clinical Centers and the 9 other sites, there's 54 participants have been recruited. It's a 2-year study, but it's a 2-year study with a quotation mark, with an asterisk, that everyone will reach the end of the study when the last person has reached their 2 years. So basically, some people that were enrolled 1 year before the last person will actually go to 36 months, and then some that were enrolled 6 months before the last patient in the study will go to 30 months. And we're relying on those extension for everyone to have a little bit more power to see a difference. At the NIH, we've been lucky enough to see 18 different participants from all across the world. We had some budget to bring people, so we've had people from almost every continent actually come to the NIH and travel every 6 months. We recognize it's a lot of travel. It's a lot for patients. It's a lot for their families, because most often they come with someone. So we're very thankful for all of them. It's expected to be completed by May 2025. So hopefully in a year, we'll have a bit more of the results. That's to give you an idea of the study design. The main point of interest that we have this time is the QMA. So it's really the muscle strength. Because of that, one of our main inclusion criteria was that 10 to 65% inclusion rate, predicted strength on QMA to be really in that very sharp slope and be able to see a difference in that limited period of time. 2 years seems like a lot in terms of waiting, but it's really not a lot when we're trying to see a statistical difference. But we're also doing a lot of questionnaires. We're doing other assessments, 6-minute walk tests, AMAT that I talked a bit before. So we do have other assessments and continue to assess safety.